Now I'd like to introduce today's speaker, Daven Presgraves. Daven is Dean's Professor in the Department of Biology at Rochester University. Daven has broad interest in evolutionary genetics. His research focuses on three areas: speciation, segregation disorder, and meiotic recombination. His work has contributed to the current understanding of sexual selection, meiotic drive, and the X chromosome's evolutionary importance. His work has led to the confirmation of a phenomenon called the large X effect, which describes the integral role of the X chromosome as a wedge in driving speciation. Daven, thank you for participating in a webinar series. Really glad that you're here. I'm going to give you the floor now.
Yeah, thank you. Thank you very much. Okay, so I'm going to talk about selfish genes. I'm going to talk about speciation, and these two things are connected to one another because of the prominent role of sex chromosomes in both of those areas. And so I'm going to start from the speciation side of things, because that's how I ended up working on the particular genetic conflicts, sex ratio, meiotic drive story that I'm going to tell you about today. And for just some context on that speciation work, I just wanted to give some, a sampling of quotes from about speciation and speciation genetics, starting right after the publication of Darwin's Origin of Species, where Darwin's bulldog, Thomas Huxley, says that there's in fact one set of peculiarities which the theory, Darwin's theory of evolution, is not wholly competent to explain, and that's the sterility of the offspring of certain species when crossed with one another. Bateson, 1922, so the champion of Mendelian genetics, says the particular and essential bit of the theory of evolution concerned with the origin nature of species, really mysterious. And then into the 70s, into the 80s, Mathematician A, Dan Hartle, and Andy Clark, same versions of the same thing. We know virtually nothing about the genetics of speciation. We're at point zero. It's an old and unsolved problem, et cetera. Okay, so that's where things stand in the mid-1980s, late 1980s, as an assessment of our field by these luminaries in our field. And it's in this time frame that something of a renaissance happens in the genetics of speciation, which I'm really credit to Jerry Coyne and to, at that time, his graduate student, Alan Orr. They call attention to two phenomena that appear to be quite general. And those two phenomena offer an opportunity, so phenotypes that are available to focus the genetic analyses. So the first observation is a comparative pattern known as Haldane's rule. And Haldane's rule, first noted in 1922 by J.B.S. Haldane, is just the observation that when you cross two species, if you find that one of the hybrid sexes is sterile or inviable, it tends to be the heterogametic sex, or in Drosophila, the XY sex, or in mammals, the XY sex. So in my table that I'm showing you here, just so you can read that, there's 137 total different Drosophila hybridizations that produce unisexual hybrid sterility or hybrid inviability. And of those 137, 131 of those are male restricted. So that's the XY sex. Similarly with mammals, it's 26-1. And of course, that pattern flips when you look at birds or lepidopterans, where of course it's females that are the XY or ZW sex. And so that finding, Haldane's rule, tells us that there's something special about sex chromosome genotype in hybrids. It's not sex per se, but rather, are you the heterogametic sex? And then the second pattern comes from classical backcross analyses of hybrid sterility. And here I'm showing you a figure from Alan Orr's PhD work. This is a cross between a backcross analysis of hybrid sterility between Drosophila pseudoobscura and Drosophila persimilis. But those details don't really matter because this pattern is quite general. You make fertile F1 females, you back cross to one of the species. In this case, we back cross to the white species, and introduce foreign autosomes, foreign chromosome 4, foreign chromosome 3, foreign chromosome 2, and so on. And then do some measure of the fertility of those back-cross hybrid males when introducing foreign genetic material. And as you can see, as you introduce foreign autosomes, fertility gets knocked down. But this male right here that I'm pointing to is a male that has a foreign X chromosome. And you can see that those males have no fertility at all. And so that male, and indeed all the males beneath him, all those genotypes beneath him, all have that foreign X chromosome, and all of them are completely sterile. That is what we mean when we say the so-called large X effect. So that large X effect is a further indication that sex chromosomes may perhaps play a special role in the evolution of hybrid sterility. But I must tell you, I was never, as a student years and years ago, was never quite convinced by these large X effect back-cross analyses. And the reason is that when you evaluate the effect of the X chromosome here, you are looking at the effects of a hemizygous X chromosome in a foreign genetic background versus the heterozygous effects of foreign autosomes in another genetic background. And so it's always possible that the alleles that cause hybrid sterility may be on average recessive. And if that's true, then of course you would expect the X to have a large effect, but it's nothing to do with the fact that it's an X chromosome. It's the fact that we're doing sort of an apples and oranges contrast here between hemizygous versus heterozygous genotypes. And so I was never quite convinced by that. So about a little over 15 years ago, therefore, I turned to the species of the Drosophila simulans clade. J.P. Masley and I did some so I should introduce the species, the Drosophila simulans species clade, which is these are species that are closely related to Drosophila melanogaster, Drosophila simulans, Drosophila seychellia, and Drosophila mauritiana. They split from each other nearly simultaneously about 240,000 years ago, and any crosses you do between them give you sterile hybrid males but fertile hybrid females. Okay, so these species, simulans is from Madagascar originally, Seychellia is from the Seychelles archipelago, and Drosophila mauritiana is from Mauritius. Now, the fact that you can make hybrids between them, where the males are sterile and the females are fertile, those fertile females allow us to move genetic material between species. So J.P. Masley and I used that opportunity to move chunks of Drosophila mauritiana into a Drosophila seychellia background. And I'm going to suppress some of the details here because I'm going to show you more genetic analyses later. We move it into Drosophila seychellia genetic background by recurrent backcrossing. And the key here is that once we, after 15 generations of recurrent backcrossing, we have a small autosomal segment, we make it homozygous in the Drosophila seychellia background. And that allows us to compare the homozygous effects of autosomal segments to the hemizygous effects of X-linked segments. It's a fair comparison. Dominance is not a confounding factor. And what we find, and I have to tell you, to my surprise, is that indeed the X does have a higher density of hybrid male sterility factors than the autosomes. It's on the order of 3 or 4 times higher, at least to the level of resolution of that study that we published in 2007, suggesting indeed the X is special and accumulates incompatibilities, hybrid male sterility factors faster than the rest of the genome. And then a second study comes from 2012 where Dan Garrigan's lab and my lab sequenced the genomes of Drosophila mauritiana and Drosophila seychellia and combined that with some Drosophila simulans genome sequence data. And what we did there is use the whole genome to make an inference about the speciation history among these 3 species. And what we find is that the best fit speciation history for the bulk of the genome is that these species split very nearly simultaneously about 240,000 years ago. But what we can do is then scan the genome and look for aberrant genealogies. We can look for genomic segments that have histories that are statistically inconsistent with that 240,000 year ago divergence time. And that's what I'm showing you here on the right. These are just little windows in the genome that have times to common ancestry that are much more recent than 240,000 years. And those are candidates for being having histories of natural introgression or gene flow between species. The main thing I want you to take from this figure is that, one, there's evidence of gene flow between all three species pairs, part one, and part two is that if you just look, you know, at the density of these tick marks, you can see that the autosomes have many more putative natural introgressions than does the X chromosome. And so putting these two things together suggests that the X chromosome is special. It accumulates genetic incompatibility faster than the rest of the genome, and that matters for the population genetic history of speciation, in that those incompatibilities may well impede or reduce the possibility, the exchangeability of X-linked segments, haplotypes. So those one may in fact give rise to the other pattern. And I will also say that this dearth of introgression on the X chromosome seems, you know, you might even be tempted to offer it up as a third, quote, rule of speciation, because it too is a quite ubiquitous finding, whether we're considering humans and Neanderthals or birds or Lepidoptera. You see the same pattern among species that have histories of gene flow that has reduced introgression on sex chromosomes. So the question I want to turn to is, why does the X chromosome behave differently? Why does it accumulate hybrid sterility faster than the rest of the genome? There is a longer list that I'm showing you here, a long list of hypotheses for why that might be, and I don't want to, and you don't want me to review all of them. So I'll just mention a few of them. One, of course, is the possibility that the X just diverges faster than the autosomes. Another is that the way the sex chromosomes are regulated in germlines is different than the autosomes and perhaps susceptible to disruption in hybrids. Another is that there's elevated levels of gene traffic, genes moving on and off of the X chromosome as species diverge from one another, which there's also evidence for that. Now, the connection between any of those three and the elevated levels of hybrid sterility on the X is tenuous, I would say. What I want to focus on today is this last possibility that recurrent bouts of genetic conflict between sex-linked segregation distorters or meiotic drive genes and their unlinked suppressors give rise to these recurrent bouts of conflict that lead to an accumulation of divergence on the sex chromosomes. This is, as I'll explain in a minute, an idea that really was championed in 1991 by Steve Frank and by Lawrence Hurst and Andrew Pomianowski. To unpack that idea a little bit, this is how it would work. The first thing we have to acknowledge is that sex chromosome transmission is evolutionarily unstable, in the sense that sex chromosomes are susceptible to invasion by mutations that can do the following trick. So let's allow a mutation to arise. So here's my X chromosome, here's my Y chromosome. Just allow a mutation to arise on the X that can do the trick of, during spermatogenesis, effectively producing some toxin that can bind or disrupt Y-bearing spermatids. So it can recognize the Y, perhaps because of sequences on the Y, and then target those Y-bearing haploid spermatids for disruption. So a fraction of them are incapacitated or killed. And as a result, there are few consequences that might follow from that kind of mutation. And so the first one is that the fertility of a male with this genotype will be reduced to some degree, because we're killing off some fraction of sperm. The other is that by killing off predominantly Y-bearing sperm, that means that there will be an excess of X-bearing sperm being transmitted, which means that we will get an excess of daughters being produced, being sired by males with this genotype. This is a phenotype that we call sex ratio, and so that's where the word sex ratio meiotic drive comes from. And then the last consequence, the last proximate consequence here is, of course, a mutation like this has a massive transmission advantage. That is to say, compared to a wild type allele, rather than sharing transmission, getting into 50% of progeny, it's getting into far more than 50% of progeny. And that means it will leave more descendants. It will spread in a population. So let me just show you a simple population genetic model of what those kinds of dynamics might look like. The lines I'm showing you here come from really simple deterministic equations where we're going to track population frequency on the y as a function of time on the x-axis here. And I'll ask you first to focus on the blue line. That'll be our DX mutation that has an, as I said in the previous slide, an 80% transmission advantage. If you introduce that kind of mutation at low frequency, it will zip to fixation in no time inside of 100 generations. The red line represents the frequency of females in the population. That proportion of the population that's female is 50%, but of course, as a sex ratio distorter spreads, the sex ratio becomes biased, the population sex ratio becomes biased, and this peaks at about 80.8. And the reason it peaks and then goes back down is because in this simulation, I'm also introducing an autosomal suppressor that has no particular fitness effect. It does one thing, that is, it suppresses drive. Okay, it suppresses segregation distortion. So when coupled with DX, it restores the transmission to 50-50. And you'll notice not much happens early on, and then it begins to spread. The reason it's spreading is good old-fashioned Fisherian sex ratio selection. So in a population that's 80% female, males who have the DX driver and an autosomal suppressor, instead of producing 20% sons, will produce 50% sons. That is to say, they will produce an excess of the rarer sex, and that gives a fitness advantage to this autosomal suppressor, allows it to be increased in frequency. So the reason I wanted to walk you through this is two main reasons. One is I wanted to show you that the dynamics of this kind of system are quite fast. As I said, we have fixation within 100 generations, and depending on the dominance of your autosomal suppressor, that goes to high frequency on the order of a few hundred generations as well. And then the second is that these kinds of dynamics, these kinds of systems can give rise to what are known as cryptic sex ratio drive systems. And the cryptic here means that species can harbor drivers and suppressors, and we might not know it. Okay, so that it remains an open question of just how common are these kinds of sex ratio, cryptic sex ratio drive systems. And part of the problem is that if, as an evolutionary, as a biologist, you come along and you study your favorite organism at, say, generation 400, you'll be tempted to conclude that there's no sex ratio distortion in your population, everything's 50-50, there's nothing to see here. But what you won't know, what you perhaps can't know, is that indeed your population has a history of drive and suppression. Your driver might even be fixed in the population. There might be some high frequency suppressors. Unless you really bash that system genetically and pour effort into looking for these things, you may well not find them. And so in the species that I'll be talking about today, Drosophila simulans and some of its relatives, we know that these kinds of cryptic drive systems exist. There are multiple cryptic, genetically separable cryptic sex ratio systems in simulans. And I'm going to come back to those in a few, at the end of the talk. Okay, so what does this have to do with speciation? So this is where the Frank and Hurst and Pomianowski model comes into play. So imagine we have an ancestral population here that becomes partitioned into two geographically isolated populations. Then imagine in one of those populations, a driver arises and elicits the evolution of resistance to drive on the Y chromosome, perhaps elicits the evolution of autosomal suppression. And now we have a cryptic drive system in one species. That could happen one or more times. Maybe there are multiple cryptic drive systems. And the same thing can happen in the other lineage as it's accumulating its own independent systems of drive and suppression. Now, when we come along and we make hybrid males between these species, there's a couple possible outcomes of these recurrent histories of drive and suppression. One is that recurrent bouts of drive and suppression can simply drive the divergence of genes that contribute or mediate or regulate in some way spermatogenesis. And it could just be the divergence in genes regulating spermatogenesis gives rise to genetic incompatibilities between species that undermine spermatogenesis. But then there's sort of more exotic potential scenarios where you can imagine that what happens when you combine drivers multiple potential drivers in the naive genetic background of an F1 hybrid, you can easily imagine where you can get aberrant or even transgressive expression of cryptic drivers in a way that leads to hybrid sterility rather than drive itself. So that's also one possible scenario. And there are others, but they're all rather speculative right now, so I'll just leave it there. So this model, the Frank Hurst-Palminkowski model, I'll just tell you that I initially found this model to be rather fanciful. It's pretty complicated. It's the kind of thing that armchair evolutionary biologists can dream up in their imaginations. But I had my doubts about all of the assumptions and how complex it seemingly is, and whether it could really be a plausible model for the evolution of hybrid sterility. But the fact is, we don't really have much data on the cause of hybrid male sterility. We have only 2 genes now that are X-linked hybrid sterility genes. The first one was discovered in 1998 in Chengyi Wu's lab by Chao-Ti Ting. This is a very famous gene known as Odysseus. And Odysseus differs between Drosophila mauritiana and Drosophila simulans in that it has experienced an extraordinary rate of amino acid divergence between those species. And then the second X-linked hybrid sterility gene is a gene called overdrive. And this is a gene that was discovered by Nitin Podnas and characterized by Nitin Podnas and Alan Orr's lab. This is a gene that causes F1 hybrid male sterility. It is also a sex ratio distorter. It causes meiotic drive. It causes biased transmission of the X over the Y chromosome. So this is precisely the kind of gene that Frank and Hurst and Palminkowski are imagining in their scenario. So here we have a sample of 2 genes. One fits, and while I'm not going to indulge right now, if you want to ask me at the end, I can tell you a story about why Odysseus is quite plausibly also has a history of drive, although that remains to be determined. Okay, so what I want to talk about today are 2 stories. I'm going to tell you about the role of drive during speciation between Drosophila simulans and its relatives, and tell you a second story about some discoveries we made recently about cryptic sex ratio drive in the simulans triad of species. So the first topic is a big project that Colin, Nickeljohn, and I did together. Our labs pooled our efforts. It's a massive genetic effort coupled with some population genomics. And so our goal is not especially imaginative. We thought, well, there's only 2 X-linked hybrid sterility genes. If we want to really know if drive if we want to assess the role of drive, we're going to need more than 2 X-linked hybrid sterility genes. We have to go get more. Therefore, we have to do a lot more genetics. And it was with no, small amount of hubris that we approached this problem at a time when we thought we have a lot more genomic resources with a lot more genetic tools. We can really control our genetic backgrounds in a way that's really going to allow us to make progress on this problem. So let me show you, I'm going to show you 2 slides of crosses to give you an idea of the mapping scheme that we use to try to get at hybrid sterility genes. And I'll just say right from the outset here that if you don't want to follow along with the details of the genetic cross, it's perfectly fine. I'm going to do my best to just highlight the key things you need to know. So in my representation here, in my schematic here, these are 2 X chromosomes in Drosophila simulans. These are 2 autosomes in Drosophila simulans. This is an X and a Y in Drosophila mauritiana and a representative pair of autosomes in Drosophila mauritiana. These are both in a white-eyed genetic background. These red triangles represent P elements, each of which carries a white plus gene, so conferring red eye color. And indeed, we can tell flies who have 1 P element versus 2 P elements. So we do this cross, we make F1 females, and then we do recurrent back crossing for 40 generations. And the idea of doing it for so many generations is to clear out the genetic background, make sure it's completely simulans-like, and to winnow away at the edges of these 2P, what I'm going to call 2P introgressions. So at the end of our 40 generations, we will have a 2P element marked 2P mauritiana introgression in an otherwise simulans genetic background. And we've made these 2P introgressions for 1, 2, 3, 4, 5, 6 intervals that together tile across most of the length of the X chromosome. So that's part 1. Then we're going to take those. Oh, and I should say, when you assess the fertility of those 6 2p introgressions, all of them cause complete hybrid male sterility. So there are at least 6 hybrid male sterility factors that need to be mapped and identified here, at least. So how do we get at them? If there's some hybrid sterility factor in the interval, we need to break up that interval and map the thing inside causing the hybrid sterility. And so we take our 2p, we're going to do another set of crosses. We take our 2p females, we cross them to simulans males that have a YFP, so a glowing eye color marker. Here's a YFP male down here. In fact, here's a 2p male and a 1p male, if you're interested in what those phenotypes look like. So we cross these two, and here's the key females for our experiment. 2p females, heterozygous over YFP, and what we're going to do from this female is recover 1p YFP recombinant chromosomes, because in those flies, we know there's been a recombination event, the 2p elements have become separated, and we're using our YFP to capture particular recombination breakpoints, because for a single female, 1p YFP female, we'll then take her and mate her to simulans males, and we can generate effectively nearly clonal males. And so we wanted to be able to generate replicate males of the same genotype, because hybrid male sterility is famously a bit of a squishy phenotype characterized by incomplete penetrance. And so we wanted to not be fooled one way or the other on fertility. We wanted to get quite confident ideas of what the phenotypes are associated with each genotype. Okay, so I told you we've got 6 2p intervals. For each, or across those, we did between 83 recombination events to 171 recombination events, depending on the 2p interval that we focused on. And for all of those, we generated at least 10 males and phenotyped at least 10 males. Okay, so that's the phenotyping. And the very last step is we use multiplex shotgun genotyping to do light sequencing of representative males from each of those classes. So we end up having 439, after we do some quality filtering, 439 introgressions that we characterize by basically doing light sequencing. So here's just part of that workflow. So we extract DNA from individual males, we use barcoding and multiplexing, sequence them, and we sequence those males to a coverage that's about 1x, and then recover the sequences, parse them by individuals, and then infer ancestry. And so the last step here, infer parentage. So this panel that I'm showing you here shows that as you go along chromosome 2, all of that red means this particular male has all simulans material. If you go across chromosome 3, all simulans material. When you go to the X, it's mostly simulans except for a chunk here that's assignable to Drosophila mauritiana. So that's one male that has a mauritiana introgression on the X. And then I just wanted to show you a couple other examples of other introgressions on the X that are of different size and different locations, et cetera. Okay, so that's the idea. And as I said, we do that for 439 males. And remember, for each of those introgressions, we have replicate characterizations of the phenotype, hybrid male sterility phenotype that goes with it. So we know something about whether they produce any offspring or how many they produce and so on. So here's the overall map that shows gives you an idea of the distribution of the fertility of these genotypes, as well as the coverage of these introgressions that really saturate the X chromosome. This is a bit hard to look at and extract information from, so I'm just going to distill all of the information you see on your screen now down into a single figure like this. On the y-axis on this side, the dotted line that you track here is going to be the proportion of males that are sterile, so the proportion out of the 10 phenotyped males that are completely sterile. And then the solid line is just the average number of progeny produced in our assay. So here, just to make sure to be clear, the axis is flipped. So up at the top means you make close to 0 progeny, and at the bottom means you make close to control levels for our assay, about 60 progeny per individual male. So what you can see is that they're about 2/3 of the X. So most of the proximal part of the X, this region here and this region here. Most of the X has a quite strong effect on a reduction in the fertility of hybrid males. And then for another third, there's 3 different regions. So the tip of the X right here, and then this middle region, there's a reasonable, there's a reduction in the compared to controls of the fertility of those hybrid males, but it's not quite complete hybrid sterility. So perversely, while I've been telling you that our goal is to study hybrid male sterility, I'm actually going to flip around things now and talk about one of these windows where hybrid sterility seems to be not comparable to the flanking regions around it. So let's focus on this window here, because when I first saw this plot, it reminded me of a different study altogether that was done several years earlier. This is a population genomics analysis we did in Drosophila mauritiana, where in about the same region of the X chromosome, we saw a similar kind of valley. In this case, though, the variable we're measuring is nucleotide diversity. Here, what we found is that as you go along the X chromosome and you look at the level of genetic variation along the X, there's this huge reduction here in the level of diversity on the X chromosome. And that is a canonical signature of selection, or that a non-neutral mutation has moved through the population quite recently, causing a hitchhiking effect that reduces diversity around it. And if you zoom in, because this is a whole chromosome scale, if we zoom in on this region, what you'll notice, again, still looking at diversity, what you'll notice is that diversity is reduced in the windows, in a sliding window way across this region. And in that region, there are a bunch of genes, but quite conspicuously we knew that there's a gene here called Dox. And Dox stands for distorter on the X. This is a gene that was first characterized by Yun Tao in 2007, which I'll say more about in a few minutes. It is a cryptic sex ratio distorter. And so at that time, we thought, Dox is certainly a good candidate for being a cause of this selective sweep. As part of our study of speciation between simulans and mauritiana, we also did a population genomics analysis, so sort of expanding from our single genome analysis to 20 genomes in simulans and 10 in mauritiana, using diversity to have more powerful study of recent introgression between species. And we used a method that allows us to detect foreign haplotypes that are at intermediate or low frequency. So quite new introgressions from one species into the genomic background of the population of the other. And the regions that show a significant deviation from what you might expect from allopatric speciation, that is the no gene flow, no introgression case. The regions that stick out are those highlighted in blue here. You can see on chromosome 2L and 2R, there are several regions that are consistent with historical introgression. And on the X, there's but one region. There are multiple dots here, but they are contiguous. So there's one interval that shows recent introgression, and you guessed it, it's the same region that spans both Dox and the gene that gave rise to Dox, which is known as mother of Dox. Again, that was something characterized by Yun Tao. And let's just focus on Dox for a moment, because what you'll see here in green are Dox adjacent sequences that come from Drosophila mauritiana. And then in the red triangles, those are Dox adjacent sequences that come from Drosophila simulans. And what you can clearly notice, one is there's evidence of a sweep in mauritiana in the sense that all of these sequences are virtually identical. But it's curious that simulans has a bunch of sequences that are identical, not only to each other, but to the mauritiana haplotype, and different from the other simulans sequences. So that's the evidence. That's consistent with this region, including Dox, moving from one species to the other in recent history. So putting all this together, it's hard not to tell this story, which is that we believe Dox originated in a Drosophila mauritiana. It caused a selective sweep, going to high frequency, eliminating variability as it went to fixation in Drosophila mauritiana. It migrated into Drosophila simulans, where it so far at least has caused a partial selective sweep in the Dox region. And then we come along and we do a genetic mapping experiment, and there's relatively reduced hybrid male sterility in precisely this region. And so that got us thinking that the model in which drive contributes to the evolution of hybrid sterility has an implicit assumption in it, which is that species are strictly allopatric, that there's no opportunity for gene flow between them. But we know that's not true between these species. If there's an opportunity for gene flow, then of course, it's not hard to imagine that those selfish genetic elements, including sex ratio distorters, can move from one species to another, as I've just shown you, clearly has happened between mauritiana and simulans. And now we come along, naive evolutionary biologists who want to genetically map hybrid sterility, and we take this blue haplotype and we move it into this species, and lo and behold, it doesn't cause any hybrid sterility. And of course, it wouldn't if they have experienced recent introgression in that region recently. Now, there's two possible interpretations. One is this is a region that didn't have much hybrid sterility to begin with, which is precisely what by chance happened to allow Dox to move between species. But there is an alternative, which is there was incomplete hybrid sterility in the region, but the transmission advantage of Dox was sufficient to overcome any hybrid sterility effects or recombine away any flanking hybrid incompatibilities that allow Dox to spread from one species to the other. And in so doing, in introgressing, it would have wiped away any divergence that built up in that region of the genome. And so in effect, gene flow allows these drive elements potentially to move between species and effectively reset any kind of divergence clock that may have been building up since this initial speciation, and therefore reduce the possibilities for hybrid sterility in the region. So it really highlights this implicit assumption that no gene flow. Story that I wanted to tell you today. And then the second story I wanted to tell you is that how our work on Dox has led to sort of a spectacular discovery that the Dox system is far more complex than we anticipated, probably than anybody anticipated. And so this is work done with a research associate in my lab, Christina Muirhead, which we published last year. And I'll just mention that a couple months later, Jeffrey Vadanayagam, who is in Eric Lai's lab, published more or less the same story because they're working on this system independently of us, made the same set of discoveries effectively. And so I'll tell you about what we find there. So I have mentioned that there are 3 genetically separable cryptic drive systems in Drosophila simulans. One is known as the Paris system. This is a system that has been characterized by Catherine Monchon-Moreau's group for over 20 years now. Really beautiful work. So if you're interested in this topic, I'd encourage you to check that out. A second system is the Durham, what's known as the Durham system. This is a system discovered by Yun Tao in 2001. And at that time, he was studying hybrid male sterility on autosomes. And what he did is he moved a piece of chromosome 3R from mauritiana into a simulans genetic background. And incidentally, without necessarily looking for it, he removed a gene called TIMI, or which he called too much yin. So I'm going to call it TIMI. And in knocking out this simulans gene TIMI, he released cryptic drive. That is, he saw sex ratio distortion caused by a driving X chromosome, but doesn't know what's causing the drive. That is to say, we don't know the identity of the drivers in this so-called Durham system. And then Dox is part of what's known as the Winter system. And as I said, this system was also discovered by Yun Tao in 2000 and described in 2007. And back-to-back papers in PLOS Biology, which are sort of groundbreaking tour de force papers in this field, I would say, where in one, he reports the discovery of NIMI, the autosomal suppressor of the driver Dox, which is described in the second paper. So let me just give you a quick summary of what Yun discovered. So first, as I said, he discovered the driver Dox. He found it was a chimeric sequence, but a quite puzzling sequence with seemingly limited coding capacity. He noted that it was flanked by these satellite DNAs that are 359 base pairs long. He also characterized the NIMI sequence and noticed that NIMI shows extensive sequence homology with Dox, and in fact shows that NIMI is a retro duplication of the driver Dox. So quite fascinatingly, the suppressor is a derivative of the driver itself. Further, beyond that, though, he noticed that NIMI has inverted internal repeats in it, leading him to predict that it ought to form a hairpin RNA. And so here is his figure of Dox messenger RNA. And here, the antisense direction here, we have the NIMI mRNA, and the green here, the IR prime and IR prime 2 are the two inverted repeats that lend themselves to the formation of the stem of the hairpin loop structure of that predicted hairpin RNA. And he predicted, well, that's a double-stranded RNA, and that may well be the substrate that leads to production of small interfering RNAs. He speculated NIMI, the autosomal suppressor of Dox, does so by RNA interference. And that prediction was confirmed by work from Eric Lai's lab that shows that indeed NIMI is a source for endogenous small interfering RNAs that indeed map right onto, of course, they come from NIMI, but they have extensive sequence identity with Dox. Okay, so that's the story, the simplified version of the Winters system. One of the things I have to mention, though, is that because of the satellite repeats that flank Dox, and because of the extensive sequence homology between Dox and NIMI, attempts to include them in genome assemblies based on short read sequence data, or indeed even Sanger-based sequence data, haven't worked out. That is to say, these genes are missing from those genome assemblies because of their sequence redundancy and because of their proximity to repetitive DNAs. And so, because they're missing, certain aspects of that story were not fully appreciated until what I've been calling the PacBio working group. This is Colin Micklejohn, Amanda Lara-Quente, Jeffrey Vadanayagam, Christy Montooth, and J.J. Emerson. Young investigators pooled their resources at the time, and they did really deep coverage, long read PacBio sequencing of simulans, mauritiana, and Seychellia genomes. And of course, those kinds of long read based assemblies allow one to resolve complex repetitive regions of the genome, including those regions that are include or flanked by these repetitive satellite DNAs. And so these 359 base pair satellite DNAs are sometimes referred to as euchromatic islands because they're sort of distributed along the length of the X rather than occurring in blocks and as is typical for satellite DNAs near pericentromeric regions. And 359, this 359 family of satellite DNAs happen to be enriched on the X chromosomes of melanogaster simulans, mauritiana, and Seychellia. This is a figure that I pulled from John Sproul's paper. This is from Amanda Lara-Quente's lab, showing the density of 359 repeats along the length, the euchromatic length of the X. In quite beautiful work, Vicki Meller's lab has shown that these satellite euchromatic islands play a role in X chromosome recognition during somatic dosage compensation. So males have one X, the dosage compensation complex needs to recognize the X. These satellites play a role in that process. Okay, so I'm going to zero in on a one window here, which is where Dox resides, and blow it up for you here. And so this is the Dox containing region going from, on our coordinate system, 9.4 to 10.4, so a 1 megabase pair region. Here is the distribution of 359 base pair euchromatic repeat islands. And in simulans, here's where Dox lives. It lives among one of those islands. Here's mDox, but to our surprise, we found that there are three other sequences, which I'm just going to generically refer to as Dox-like sequences because they are clearly related to Dox and mDox. So there are more than just Dox and mDox. There's three additional ones. And then in mauritiana, we found 12 more. And in Seychellia, we found 12 more. So Dox is really only one member of an expanded family of Dox-related sequences. There are 5 in simulans. There are 12 in mauritiana, another 12 in Seychellia. 8 of these are unique to a genome to one of these species, so they seem to be species specific. Another 8 are present in 2 of the 3 species, and there are only 2 that are present right here in all 3 species. So I told you that Dox has chimeric origins and limited coding potential. So with this whole gene family, we're able to reconstruct, sort of forensically reconstruct the history of these, the building up of the Dox sequence. And there is an initial sequence which is derived partly from the gene tapas and another gene called protamine AB, which inserted into a gene called CG8664. Let me just pause here and say, do not bother yourselves trying to follow the details. Rather, just appreciate how complicated what I'm about to explain to you is. The names of these genes, except for one of them, is not critical at this time. So one, we have a fragment. We don't know exactly where that came from, but it inserts into CG8664, which is on the X chromosome. A piece of that gene that spans protamine AB and CG8664, then inserts into a gene called CG15306. CG15306 is a testis expressed gene, and this sequence effectively replaces the coding region of the gene and leaves the 5' and 3' UTR intact. And we call that gene Dox because we think that's the source sequence that seeds the expansion of the Dox-like gene family. The reason we think that is because the very next insertion event involves a piece of Dox inserting into a 359 euchromatic satellite DNA island, and we call that Dox-like. We call that first sequence Dox-like 1, in fact. And so here in dark green is where Dox lives. You'll notice there's no satellite DNA there. Then here's Dox-like 1, which is associated with these 359 satellite DNAs. And after Dox-like 1 is founded, all of the other Dox-like sequences, as we move from right to left or proximal to distal here, all of them insert into pre-existing satellite 359 satellite DNA islands. So why might that be, or why should that be? And this is speculation, but I think, reasonable enough, which is that you can imagine these satellite DNAs present paralogous sequence homology. They present targets where if you have, for example, a double-stranded break in one satellite DNA, it can template off of a paralogous sequence for repair and incidentally be converted. The Dox-like gene here can become deposited at this other paralogous position. That's one mechanism that you can imagine. So paralogous conversion of satellite DNA islands. And then there's another mechanism for which we definitely have evidence, which is that you can imagine that the satellite DNAs that are flanking here can actually template or pair among themselves and allow for loop outs to occur. That is the production of extra chromosomal circular DNAs, which can then, once again, via sequence homology pairing with other satellite DNA, paralogous satellite DNA islands, have a recombination event and pop in a Dox-like gene at another site, another satellite 359 island. And as I said, we have evidence of that, but I'm not going to show you of that kind of event, but I'm not going to show you that. Okay, so let's turn now to Dox, or MDox and Dox. Where did they come from? And I'll just quickly walk through their histories, because once you have Dox-like sequences, a Dox-like sequence then inserted into a satellite 359 island that's adjacent to a gene called cubulin, creating MDox, and then MDox plus some of this cubulin sequence inserts into yet another satellite 359 island, and that's how Dox is created. And so I wanted to walk you through that. Again, I'm not asking you to keep track of all the genes involved, but rather the complexity. That is to say, there are 5 different insertion events that led to the production of Dox, and that's just one. I'm sure we could unpack the particular histories of insertion and evolution of the other sequences and find similarly complex stories. So this is 4 different sequence origins, 5 different insertion events giving rise to the production of the Dox gene. All of these Dox-like sequences, when compared and taken in aggregate, reveal a history of rapid structural and rapid sequence evolution. So here is a schematic of how it looks when you align these sequences from simulans, mauritiana, and Seychellia. You can see lots of insertion and deletion events showing that indeed there's been some structural evolution there. And then if you look at sequence diversity among all these copies, the thing I want you to notice is that one, of course, there's some diversity among the sequences, but in this blue highlighted bar, there's quite a lot of, or elevated levels of sequence diversity consistent with rapid sequence evolution in this window of the light blue and yellow part, especially this light blue part. And that part corresponds to the sequence that originated from a gene called protamine AB. And protamine AB, as the name suggests, protamines, if you know what those are, they are sperm-specific histones. Okay, so all of the Dox-like genes, or very nearly all but perhaps one, encode a protamine-like, a highly derived protamine-like sequence. They encode proteins that have HMG box domains. Those are DNA binding domains. And protamines are the small basic proteins that replace canonical histones during spermiogenesis. Canonical histones are stripped off and they're replaced by sperm-specific histones, which are much more compact and help to facilitate the reduction in the size of the volume of the pronucleus. It's reduced nearly 200-fold, in fact, going from pre and post histone transition. So that sperm-specific histone clearly, I should have said that there was limited coding potential, and it's true. This is a very short open reading frame, so it's not shocking that it went undiscovered by you, and wasn't terribly obvious. But what we know about protamines, obviously just being a protamine, you can already tell yourself a story about how that might lead to drive. You can imagine a protamine preferentially disrupting Y-bearing sperm. And I'll just mention that if you perturb protamine AB in Drosophila melanogaster, you can phenocopy segregation distortion as caused by the segregation distorter male meiotic drive element in Drosophila melanogaster. So this result, the protamine result from segregation distorter, may indeed be highlighting the fact that protamines and protamine adjacent sequences are spots of vulnerability during spermatogenesis. Transitions or functions that are susceptible to manipulation by selfish genetic elements. So last, I showed you a lot of structural evolution, which might raise the question of, are all of these genes functional or not? That is going to require functional validation for all of them, but I can at least tell you that we used Dox-like gene-specific SNPs, so diagnostic SNPs unique to particular copies, to ascertain whether they're expressed, individual copies are expressed or not. And we find that indeed most are expressed. So 4 out of the 5 copies in simulans are expressed, 7 of 12 in Drosophila mauritiana are expressed, and 8 of 12 in Drosophila Seychellia are expressed. And I want to say that that's conservative because some of these Dox-like genes may not have diagnostic SNPs. And furthermore, when I say they're expressed, they're actually expressed in genetic backgrounds where we believe these systems are probably transcriptionally silenced to some extent. And the reason for that is because most Dox-like transcripts, most sequences with Dox-like sequence, originate and are traceable to, in simulans, to NEMI or indeed to TME. So this is a discovery made from Eric Lai's group. Again, this is that same Lin et al. Paper from 2018 where they found NEMI has Dox-like matching sequences, small RNAs that are being produced. The same is true, but they're mappable to the TME region. It wasn't known until they made that discovery that TME must therefore have some evolutionary relationship to the Winters system. Remember, TME is the suppressor in the Durham system. But one of the things we noticed is that, well, actually, I'm going to come back to that. So in Drosophila mauritiana, which also has NEMI, we discovered a gene called TME-like. So NEMI is a retroduplicate, TME is a DNA duplicate. Timi-like is also a DNA-based duplication. And in Seychellia, they also have a version of Timi-like. We think these two are non-independent. Seychellia has a further autosomal suppressor called EMI for even more yin. And that is an interesting case because it's only a 200 base pair fragment derived from a Dox-like gene inserted into a testis-specific gene, duplicated, inverted, and then a 7 kb region around those 400 base pairs and that testis-specific gene became amplified 4 times. So there's 28 to 30 kb region that's 4 tandemly amplified copies of the EMI gene, which we then validated all of those genes. So Timi, Mimi, Timi-like, EMI, and all 3 species produce small RNAs that match Dox-like genes. So one of the things we noticed is that some autosomal endo-sirRNA genes or hpRNA genes have sequence homology that match some Dox-like genes better than others. And it really does appear that in each species, there's a partitioning of function. That is, different suppressors are specializing on different subsets of the Dox-like genes on the X chromosome. So the question now is, why have multiple drive loci? Why have multiple suppressors? And so one obvious suggestion is you can imagine a driving gene arises on the X. You can imagine, and this is all just speculation now, but it's a model that can explain how we got to this complex web of drive and suppression in these species. You can imagine we have a driver elicits the evolution of a suppression system like NEMI suppressing this one. If drive is dose dependent, that is to say that making an extra copy facilitates escape from suppression or simply boosts the strength of drive, that will favor the evolution of additional copies, the recruitment of additional copies of Dox-like genes. Once you have additional copies, if they're able to escape a suppressor, that could lead to the production of tandem amplification as an EMI or the production of yet another autosomal suppressor in the case of Timi-like or Timi. And then once you have multiple drivers and multiple suppressors, you can easily imagine you now have the substrate for sequence diversification, because sequence diversification may well be another way to escape silencing by these small RNAs. And so this newly evolved sequence here may escape these 2 suppressors, which then leads to selection for one of these, either the founding of a new suppressor or adaptation of that new suppressor to then be able to suppress that new driver. And so this is how you can imagine this sort of redundant or multi-copy substrate can give rise actually to sequence diversification and specialization of suppressors on different subsets of drivers. So here's just my overall summary of the system I've just described in simulans, mauritiana, and Seychellia. There's 18 total Dox-like loci. We think the satellite DNAs were essential to their amplification and indeed gene conversion between copies. Many of these are species specific. And in fact, all of these taken in aggregate are absent from melanogaster. So in that sense, the whole family is lineage restricted. I showed you there's quite a lot of structural diversity, very rapid sequence evolution. All of them encode a protamine, and most are expressed even in a quote unquote suppressed genetic background. And then we have the origins of 4 different autosomal suppressors with 2 in each species, again, specializing on different subsets, and all 4 have independent origins. And as I showed, all of them produce small interfering RNAs. So the big picture conclusions from the talk, I hope that you take away. One is drive can indeed contribute to the evolution of hybrid sterility. I showed you the case of overdrive that was well established. But from the first part of the talk, I showed you that with even a little bit of gene flow, clearly these selfish sex ratio drive elements can move between species and even potentially undermine the evolution of hybrid sterility. And then second, I showed you a really complicated story of how the founding of a chimeric sequence that can cause sex ratio meiotic drive can then become amplified and give rise to a web of multi-copy drivers, multi-copy suppressors that are lineage restricted. And that last sort of, overall picture is something, we can talk about this in the Q&A if you want. It sounds incredibly esoteric and incredibly maybe even unique. But what we can talk about in the Q&A is that it's over the last decade alone, it's becoming clear that that is not unique. In fact, a lot of the things I've described here, as complex as they are and as idiosyncratic as they are recapitulated over and over again in different drive systems, in different taxa, ranging from fungi to mammals. So there are some general themes that are beginning to emerge in our understanding of the genetics and the genomic consequences of these kinds of selfish meiotic drive systems. I'm going to end by just revisiting this slide that I introduced at the beginning, which is, there's all this lamenting for, over 100 years about not understanding why in the world would hybrid sterility evolve in the first place, and a, throwing up of hands, because most speciation is a product of ecological diversification. It's an incidental byproduct of adaptation. But it's not really obvious what about male fertility is adaptive as two populations go to different, why should two populations evolve incompatibilities, especially quickly, that affect the male germline? It's hard to sort of construct an ecological story there. The reason I think a lot of these luminaries in our field struggled so mightily is they absolutely either didn't know about the kinds of phenomenon I just described to you. Things we're only really beginning to come to terms with and understand now. That is the pervasiveness of selfish genetic elements and their role in driving genome divergence between species. These are just, phenomena that were not on their menu when they were considering how these things might have happened. So I really think, one, the ubiquity of drive is being revealed, and two, the consequences we're about to experience a new era of understanding the consequences of these kinds of conflicts for myriad phenomena, even ultimately the origin of species. So let me just end there. There are lots of people to thank, but for the brains and the effort, I need to thank my PhD student, Emily Landine, who did a heroic amount of the hybrid male sterility work, Christina Muirhead, who, of course, spearheaded our effort in trying to understand the evolution of the Dox system, and of course, Colin Micklejohn, for him and our labs, working together to try and understand the evolution and genetic basis of hybrid sterility in these groups. And of course, the PacBio working group, who I've already referenced, because they were kind enough to share their PacBio data sets with us pre-publication, for which, of course, we're grateful. So I will end there, and I'm very happy to take any questions.
Thanks a lot, David. It's really, really fantastic and super coherent and clear, really, and impressive. So let me encourage folks to ask questions. You can either chat those in, or feel free to raise your hand, and we'll try to recognize you, and we'll open your mic. I'm just going to start with maybe a clarification. You showed this really complex pathway or sequence of movements of Dox or Dox fragments that were each of those movements associated with a functional with functional meiotic drive. Was that what was sort of keeping it all going?
Let me see. So I think the first answer to that is, let's see if I can go backwards in my slideshow here. Oops, went too far. It is hard to imagine there being no meiotic drive until Dox was formed. So we strongly think, loci other than Dox were, of course, had the capacity for drive. That's why you get suppressors evolving and so on. But to know the fine details of which events are incidental versus have functional consequences for perhaps strengthening drive or facilitating escape from suppression, that I think is hard to know without experimentally dissecting individual gene copies and perturbing these sequences, which I think we're very much at the beginning of that, trying to understand that process.
Yep. Okay.
I will mention that. I will mention that I have actually suppressed some of the complexity here in that some Dox-like genes there are what may well be passenger sequences that are part of these Dox-like genes. Some have it and some don't, and it's really, as I said, it's unclear how many of those passenger sequences are incidental versus functional. So that's an open question.
Yeah. Sarah Zander has a question. Are the 359 base pair satellites transcribed? How did they get to be distributed along the chromosome?
I'm not sure I can answer that question. So they are transcribed, although I can't. I feel a little uncomfortable saying they are transcribed because I don't know if it's a subset of them or I can't really speak to that. Perhaps somebody in Amanda's group or perhaps somebody from Vicki Miller's group could. And then how did they get distributed like that? I also cannot answer that except point you to Amanda's paper and others who show that these repeats are enriched on the X. That's been known for a while. And now Amanda's group has shown in a paper by Sproul et al. In 2020 that there seems to be some turnover of those satellites. That is to say, melanogaster has almost exclusively satellite 359 islands, and those islands have been invaded in the simulans clade by responder-derived sequences, which appear to be displacing the satellite 359 sequences. So the loci are staying the same, but they seem to be being replaced gradually by responder. The significance of that, why that's happening, I can't answer. It's one of those satellite DNA enigmas that we're going to have to wait on, I think.
Eric Lies in the audience has a question. Eric, I'm going to try to unmute your microphone here. Let me see if I can do it. Katie, if I can't do it, can you just unmute Eric's microphone?
Hi, can you hear me now?
Yep, we can hear you, Eric. Go ahead.
Hi, David. Hi. These systems are so fascinating. I have a question that maybe I'd love to get your perspective. So it makes a lot of sense that drivers are originating on the X for sex ratio meiotic drive. I'm wondering, do you know an experimental test that they have to live on the X to mediate functional drive? That is, do they have to work in cis? Could it be placed on an autosome and still mediate drive? And if not, could drivers be ever evolving on autosomes and we didn't notice them, or could X chromosome linkated drivers be functionally cooperating with some autosomal elements that maybe they weren't noticed?
Right. So the population genetic models here of male meiotic drive, the genetics and then the population genetics. The genetics is that male meiotic drive tends to be multi-locus with a minimum of 2 loci, as you know, a driver and a target. And on autosomes, the only way for a drive element to invade and spread and get a foothold in the population is if there is some initial genetic linkage with the target sequence. So SD is the classic example. The T haplotype in mouse is the other, where on autosomes, you have a driver that target a sequence that's nearby. And the idea is that the driver originates in coupling or is quickly placed in coupling with a resistant target. And then those tend to get captured by inversions or tend to reside in regions of low recombination that allows the driver to spread, because if there's free recombination between them, then you can get the formation of suicide combinations. That is, targets that come in resistant versus sensitive alleles. If you couple a driver and a sensitive allele, it'll drive against itself. And if there's free recombination, it'll kill itself as often as it doesn't kill itself and can therefore not spread. What makes sex chromosomes special in this regard as a breeding ground for drive elements is that the X and the Y chromosomes, for the most part, are non-recombining. And so in principle, any driver on the X can target any sequence that's unique to the Y without any sort of drag created or inhibition created by the possibility of recombining a sensitive target onto that same chromosome. So I don't think it's to do with function. The understanding anyway is not to do with function per se, although I can imagine possibilities. But I think it's rather, it represents a kind of sieve. That is to say, you can imagine driving mutations arising on autosomes and X chromosomes, let's just even say with equal frequency. It's just that the ones that arrive on autosomes never get off the ground, but the ones on X chromosomes can invade for the linkage reasons I just gave.
Great.
And I will mention that, in the SD system, Terry Little has these nice experiments that show that you can move the responder locus around. So you can take the responder from chromosome 2 and you can move it to the Y chromosome, and you can show that SD still drives against it. So, in principle, I guess that leads me to think that it's not a location per se. At least in that system, it's clearly the case that you can move the components around and sort of engineer drive based on where the target is.
Great. Thanks, David. Thanks, Eric, for the question. And I encourage the audience to, feel free to raise your hand and ask a question. Takeshi Akira has a question. Why do you think mauritiana and Shisheliya have more Dox-like genes than simulans?
I don't know the answer. If you force me to speculate, we know that mauritiana and Shisheliya have smaller population sizes compared to Drosophila simulans. What I can imagine, therefore, is that drivers might arise and invade a simulans population, but get silenced rather quickly. And if they get silenced and they're not able to get to high frequency, once silenced, they can fall apart. Right? If you're a silenced drive element, you have no function, there's no selection keeping you around, and you fall apart and you disappear. In mauritiana and Shisheliya, where census population sizes are smaller, it's easy for me to imagine a driver arising and being able to spread to high frequency in the population faster than suppression can evolve to stop them. And so that would be my speculation. I mentioned during the talk that Catherine Montchamp-Moreau's work on the Paris system. They have really, quite beautiful work of looking at longitudinal data on the frequency of the Paris driver and the frequency of Y chromosome resistance in the Paris driver. And that's a drive system where the dynamics are, on the order of tens of years. So, meaning major frequency changes. And so there's a case that's consistent with systems being silenced before they make it to fixation in simulans. But that is speculative. So, but it does suggest that there are other considerations here, demographic considerations to consider when thinking about the fates of drive elements.
So are the evolution of those resistance mechanisms or suppressor mechanisms that rapid? So, for example, the Dox suppressors.
I have to tell you, I'm a little torn on that because on the one hand, we have 4 independent. So I have to just say I'm quite blown away by the winter system. There's 4 new genes in existence that produce small RNAs that just aren't there in other species, and therefore almost certainly originated within the last 250,000 years. And to me, that seems quite spectacular. And so it suggests, well, maybe it's not that hard to make suppressors. On the other hand, the suppressors we're talking about here are quite complex structures. You had to have a duplication. That duplication had to have acquired an inverted repeat in order to possibly give rise to the endo sirRNAs. It has to get transcribed in the testis. So there's a lot of sort of accidents that happen along the way. And therefore, they're not single nucleotide mutations. And in a really big population, even like Drosophila mauritiana or simulans, you have to imagine that every generation, almost every nucleotide mutation possible is probably being sampled every generation in some haplotype somewhere. And so because these mutations are, on the one hand, accessible, they've been made multiple times. On the other hand, they're less accessible than simple nucleotide evolution. And we think there's reason to think when you get gene flow of a drive element from one species into another, like Dox from mauritiana into simulans, not only does that undermine the evolution of hybrid sterility, but now that there's a new driver in simulans, there's pressure to evolve the evolution of suppression of that driver. Now you can evolve suppression of that driver de novo, or you can wait for a little more gene flow and you can sample the suppressor that's already present in mauritiana and pull it over. And we think that's what happened with Nemi. We think that Dox came over, and we think, this is unpublished, but we think Nemi has been pulled over as well. So we have an entire cryptic, sex ratio cassette, Dox and Nemi, discovered, characterized in simulans, and we think both of them originated in mauritiana and were both pulled over because of the non-independence between them. The reason I'm mentioning that now is because what that suggests is that the opportunity for the evolution of suppression, that gene flow was the route to suppression rather than de novo evolution of a new suppressor of Dox in simulans. And that does suggest to me that the possibility that there these might not be immediately easily accessible, in that a more complex mutation that already exists, it's easier to sample that by some infrequent gene flow event, rare gene flow event, rather than waiting around for the evolution of a new suppressor. So there's kind of a tension there between, they clearly can do it, and yet there's a speed limit on how quickly they can do it.
Are the Paris suppressors de novo? Did they arise de novo?
I can't speak to those because in the Paris system, there's resistance to drive on the Y chromosome, but it's variable. Quantitatively variable, that is. It's not all or nothing drive, and it's also geographically variable. And then as far as the autosomes go, the Paris system has, there are attempts to map the autosomal suppression, of which there is autosomal suppression. My recollection is that it reveals that it's quite multigenic. So there's not a single autosomal suppressor, but rather multiple loci involved. And there are stories, you can speculate about why the difference between the winters in the Paris system, but I will save my speculation on that one. That gets us pretty deep in the weeds, I think.
Yeah. Pratima Chanuri has a question. Hi, Pratima. Her question is, first of all, it's absolutely fascinating talk. She loved the classical and advanced genetics employed here. Do most of the suppressors that seem to evolve on autosomes act through small RNA pathways, or are there other mechanisms? So, yeah, good. I mean, there's something I had in my mind as well. Just if you could do a little comparative look at other systems and how do you see this kind of a mechanism elsewhere?
I hope I'm not forgetting something, but I cannot think of one off the top of my head that is like this, except for circumstantial evidence that there are lots of examples of so-called ampliconic or multi-copy genes on sex chromosomes that are testis expressed and lineage restricted, if any of that sounds familiar to you, that really seem like they're good candidates for drive, and multiple examples. And indeed, some of those they're either suspected drive cases that do produce small RNAs that do silence the thing that we suspect once was a driver, or the genes themselves are enriched for functions in chromosome segregation or small RNA pathways. So both of those categories are enriched among things that we believe are drivers. The reason I have to phrase it in such a cagey way is that there are lots of cases that look like they were, or possibly are drivers, or are now defunct drivers, relic drivers, because they've gone to high frequency, and once fixed, they just died and that's it, and they've left their legacy in the genome. And so in those cases, you have to be a little cautious because it looks like a driver, but that doesn't mean it is. We don't have the experimental evidence to prove it. On the other hand, it's also quite difficult to explain many of the increasing number of observations coming off, especially sex chromosomes. Once again, it's hard to explain it without drive, I think. So I can't think. I guess I will say that in the segregation distorter system, you can phenocopy segregation distortion by perturbing the pRNA pathway. So that does suggest that there's a small RNA mediated, small RNA component to the segregation distorter system. But I don't know that how well worked out those details are. Partly because the mechanism of segregation distortion is not pinned down precisely yet.
Well, great. So we're at 12:25, just about. I'm going to. I think we'll begin to wrap it up here. What I'd like to do is just mention to the audience that the series is continuing next week. Well, before I say that, let me just say thank you to Daven again for a really fantastic talk. And some of the genetics was really a tour de force, which was really the amount of work involved in those studies was, well, I appreciated it.
Thank you. That's very kind.
Yeah, it was really something. But let me just say that this webinar series will continue next week. It'll continue back on our usual Wednesday time slot, and it will feature Anna Lindholm, who will talk about the T haplotype system. And she's been working on some very interesting aspects of behavioral ecology and the influence of the T haplotype on and how it is successful to the limit that it is in mice. So Daven, thanks again for being here today. It was really fantastic. Thanks to the audience for being here as well, and thanks for the questions. And with that, I will say good afternoon and have a good day. We'll see you next week.
All right. Thank you very much, Dave.